So we'll be getting started now. Here is a contents page where I'll just give you a brief rundown of what we'll be talking about today. So I'll start with an overview of ENA as a whole, what it is, what we do, and then we'll talk about why you would submit your data. Hopefully if you're here today, then you're already sold on that, but we'll run through it. We'll talk about the data model and metadata model, and a good understanding of these is going to be really helpful to you when it comes to structuring your data and making your submission. And then we'll kind of run through an example of how you would structure a study. So ENA is a very broad platform for archival of nucleotide data. We take all kinds of different sequences, pretty much anything that doesn't have a data control policy, anything with human identifiable data, anything that doesn't fit that category, we take. We've been running since the early 80s and have been extended since as new technologies and applications have come along. And now we are fully online as of like a decade or two ago. We're the European node of the INSDC. That's the International Nucleotide Sequence Data Collaboration. And what that means is that we share data with NCBI and DDBJ. So we all accept submissions of data and mirror it to the other two databases. So anything that you submit to one database will end up in the other two. And it's just a question of which one you prefer the submission interface of. So we provide a rich set of tools for submission and retrieval of data. We'll be talking a little bit about these submission ones as we go. And we're very much linked in with other resources around EMBL and EBI, who use subsets of ENA data in various ways to create interesting other services. As of September, we contain more than 8.5 petabase pairs from over 1 million different taxa. So our collection is really extensive, and that gives us a lot of power to do some really interesting stuff and to empower other people to do interesting stuff. At any given time, we have between 2,000 and 5,000 active submitters who come from all over the world. There's a global community of people accessing and submitting data to ENA. Submission to an INSDC database is often a mandatory step in getting your work published. So journals will often insist that your data be made public before you can publish your paper with them. So this motivates a lot of submissions. And we're just going to take a moment to discuss the motivations for submission. So I've just mentioned that it's often necessary for publication, but there are some other reasons, like, for example, MGnify analysis. MGnify is another group within EBI. They're not a part of ENA, but they work with us closely. And if you submit your data to ENA and give your consent to MGnify, then they will run your data through their analysis pipeline, and you'll get some free analysis done on your data, which a lot of people find very useful, helps give them some grounding in running the analysis. We believe that having open data leads to greater reproducibility and greater accountability. So having the data out there means that claims made in papers can be tested and verified. It's also useful sometimes to allow third parties access to your data. If you have collaborators who are very geographically remote from you, it can be a very useful way to make your data available to them, although of course it is then available to the entirety of the rest of the world. And it can be a useful means of archiving your data. So storing sequence data can have some very big overheads, especially for smaller groups. So having it archived somewhere as secure as ENA is very useful for a lot of people. So with that out of the way, let's talk a little bit about how we organize sequence data. This is our data model, and understanding this is quite useful for sort of understanding how you're going to make your own submission. So basically, we organize all sequence data into one of three tiers. So at the bottom, you have reads, and this is the raw output of a sequencing machine. So this would typically be something like a FASTQ file or maybe a BAM. There are a few different formats we accept, but it's the raw output of a sequencing machine with minimal processing done on it. The next tier up from that is assembly. So this is the result of taking those reads and finding regions of overlap using some kind of algorithm to produce contiguous sequences which represent fragments of real biological molecules like chromosomes or plasmids. You can see this builds on the reads tier directly. and we treat it as different in the database, although there will usually be a relationship between assembly and read data. Building on top of that again is the annotation tier. So this is an interpretation of biological function, and that is projected onto an assembly at a coordinate defined location. So this is not so much sequence data in the way that the other two are, but it's projected onto sequence data. For the sake of metagenomic submissions, though, we're not so interested in annotations. We're mostly going to be talking about reads and assemblies. And there are a few different types of assemblies that you might be submitting if you've done a metagenomic study. And I'm going to take a moment to explain what those are now. So the lowest level of assembly you might have done is a primary metagenome. This is the set of assembled contigs, but you've not done anything to try to separate those contigs based on taxa. There are no assumptions made about what those contigs mean or what their origin is. And then building on that is the binned metagenome. So this is, again, a set of contigs from a primary metagenome, but you've grouped them into a single taxon set. There are different criteria, different ways you can do this, but overall, it just means that you have grouped some of the contigs on some basis. And finally, the next level up from that is the metagenome assembled genome, which I'll be calling a MAG from now on. This is a completely assembled genome drawn from a binned metagenome, which you assert closely resembles a real individual genome. So this is perhaps one of the really great benefits of doing metagenomics, because it allows us to assemble and understand the genomes of species that can't be cultured through traditional means. So MAGs are a really great application of metagenomic sequencing. So just to recap those then, the relevant data types that you need to be thinking about if you're submitting a metagenomic study include reads, the raw output of sequencing machines, and assemblies, which include primary metagenomes, binned metagenomes, and MAGs. You may not be submitting all of these things, but it's important to be aware of them and to be aware of the possibility when you come to do your submission. So that covers the different types of data. We're going to take a moment now to talk about metadata. This is what gives your data context. Without the metadata, your data has no value being in a public database, because the metadata tells people how the sequence data was produced. It makes it possible to compare data sets from different users, and that's at the core of what makes ENA useful as well. But it allows people to run really complicated and interesting queries. There's an example here. I want to see data from bacteria in the Atlantic Ocean, sampled between 50 and 100 meters down, between April and July, compared with the same from the Indian Ocean. That is a query or two queries that you could run in a fairly short amount of time using the ENA advanced search tool, just because people submit metadata with their data. It's typically a requirement that they do so. And it makes data much more useful. So the way metadata works is that we have submitters register different objects that describe different aspects of their project. So if you look at the map up here, you can see the actual data is just a small part of what's going on here. It's this red box here. We build these different objects around it, which, as I say, describe different aspects of your project and make your work much easier to understand and compare for people who might stumble across it in the database in the future. So I'll walk you through each of these objects, and you can think of them as representing different stages of your project. So in the first case, you're kind of sitting in your lab or your office, thinking about what sort of new thing you want to learn, what the goals of your next study are going to be. And this kind of data will be registered with ENA in a study object. So often a good way to do this is just to, once you've written it, paste your abstract into it of your paper. It also has the function of grouping all the other things that you're going to submit underneath it, and it controls the release date of your data as well. So when you're ready to release your data, you can do it very simply just by coming along and changing the release date of the study. So the next thing you'll do with your project is go and find some samples. So if you're doing metagenomics, this could be some seawater or some soil or some samples from someone's gut. Whatever it is, you'll log various details about them in the database, and you'll make one sample submission for each sample that you have in the real world. And you will withdraw DNA or RNA from this, and you'll do some sequencing on it. And the details of how you do that sequencing are what you will log in the experiment object. So that contains things like the library preparation method, the sequencing platform you use, that sort of thing. The result of doing that sequencing will, of course, be some reads. This is your raw data, and that will be contained within a run object. And the run primarily just has the function of pointing to where the data is in an FTP directory. That's the main thing that a run does. And of course, you might then do some assembly on your read data, and you'll register that in the form of an analysis object. Analyses are very much a broad category. It can contain all sorts of things, but it does contain assemblies. So you'll be registering them if you want to submit any assemblies to us. And down the right-hand side here, you can see that everything that you register gets given an accession number, and they look like this. They all start ER, and then they have a different letter depending on what the object is. So we have P for project or study, S for sample, X for experiment, R for run, and Z for analysis, followed by some numbers. And so that can be quite helpful to know those things. If you know what an object is just by looking at the accession number, that can help you with navigating data in ENA. So now we talked a little about that. I just want to run you through how you might actually structure a metagenomic study from a metadata perspective. So in the first case, we're talking about read data submission. So this is a study that you might submit which contains a single experiment and run. So the study is over here. As I said, that describes the goals of the experiment. Down here, we've registered a sample. We can see that this has been registered as gut metagenome. That goes in the taxon field. And we have registered over here an experiment, and this contains details like the name of the library, the fact that this was a paired end experiment, the fact that it was done on an Illumina HiSeq 2000. The experiment also fulfills the role of pointing to the study and the sample. So it's actually starting to link these things together into a group. The experiment also has the run pointing at it. So you can see this describes the file names. In reality, there would be a full path to an address in an FTP directory, but I've just included some example file names here for brevity. So this is about as simple a study as you could submit to ENA. In reality, you may have many runs. You may have many hundreds of runs, but you may also have only a few. I've just used one here for simplicity's sake, but you can copy and paste this as many times as you like. Each experiment you submit would still be pointing to the same study, but might be pointing to a different sample. So now that we have registered our reads, the next thing we might want to do is register a primary metagenome. So I said earlier, a primary metagenome describes or contains a set of contigs that have been assembled from the reads, but has no effort to separate them based on taxonomy. And so that's sitting over here. It's contained within an analysis object. Remember, I said assemblies are contained within analyses, and it describes that it's a primary metagenome, and it points to the data that it was produced from. It points to the sample that it was produced from, and of course, it points to the study that it is contained within. The next thing up from there is a binned metagenome. Once again, it's contained within an analysis, and it points to the same things. It points to the run, to the study, and to the sample. So when people come and look at these things in the database later, it's going to be very clear how exactly they were made, because as well as that, the analysis is going to contain things like coverage and the program that you use to do the actual assembly. The final assembly object that we might be submitting is a MAG. We talked about these earlier. This is an actual genome assembled from metagenomic data, and you can see this complicates things a little bit more. Because the MAG actually describes a individual species rather than a metagenome, that needs to be reflected in the way it's modeled in the metadata. So you can see our mag again points to the run because it's pointing to the same data as the other two that it was made from. It's pointing to the study, of course, but it's pointing to a separate sample. And this sample has E. coli as its taxon because in this example, E. coli is what we've chosen or managed to assemble from our metagenome. It has a extra attribute indicating what it was derived from. Most samples don't have this, but when you're submitting a mag, you have to register a sample that indicates that it was derived from another sample. So now we've talked about how you would structure the different things that you would be submitting. I'm going to talk a little about the different routes of submission that are open to you at ENA. So there are actually 3 different ways that you can do it. The different routes are good for different things. Interactive submission and programmatic submission are the older ones. These have been around for a while. Webin-CLI is something new. Interactive submission is perhaps the most familiar and accessible. You just use your browser to fill out web forms which describe your work. Programmatic submission, you describe your work in XML documents and you submit those to us using a program called curl, but conceptually it's the same. Webin-CLI is a smart new submission interface that has recently been made in-house and is being released at the moment. This has the advantage of validating your files in advance, which we'll talk a little bit about as we go. Soon enough, it's going to be the only way to submit assemblies and read data. You can currently do this with interactive submission, but it's being turned off soon because Webin-CLI offers lots of advantages that we want to make full use of. So when you come to register a study, you can do this using either interactive submission or programmatic submission. So as I say, with interactive submission, you just fill out a few boxes in your browser. So in this case, we've got the release date of the study. We have a short name for it that you can refer to it by. We have a title, which you could use just the title from your paper, an abstract to describe your study. And again, you could just come back later and paste your paper abstract in here once you've written it. I just want to draw your attention to this box over here. This is the PubMed ID section. You can paste a PubMed ID in there, and then when your data is public in the browser, when people come along and look at it, there'll be a link to your paper so they can go along and read a bit more about it. You can do this, register a study using programmatic submission by preparing it in an XML document. If you're not familiar with XML, it kind of works the same as a web form in a lot of ways, but instead of containing things within boxes, it contains them within tags that say the name of the thing they contain. So in this case, metagenome study goes in this name box, name tag, sorry, and that's how you describe it. And you can get a template of this from our tutorials, and so you don't have to somehow know how to put all of these things together. As far as submitting XMLs goes, it's a fairly simple procedure. You prepare a second XML called the submission XML, and that is primarily just to tell the system what to do with the other XML document that you're sending it. So in this case, we're telling it, add the thing that I'm sending you to the database. And then you submit it with a command line program called curl. If you're not familiar with command line programs, the way they work is you just type the name of the program you want to use and then give it a list of options. So in this case, we're giving it the U option, which tells curl that you're giving it your username and password. So these are your password and username for what we call our Webin client. You give it the locations of the files, so the submission.xml, which is this one, and the sample.xml or study.xml in this case. And then you give it the address you want to send it to. And it's as simple as that. Curl will print out a message letting you know whether your submission has been successful or not, and it will give you your accession numbers, and you'll be ready to move on to the next stage of your submission. I just want to call out this slight quirk in the URL here, www.dev. This means that you're submitting to our development submission service. And all that means is that it's a practice test submission, and it will be removed after 24 hours. So if you're not comfortable with doing your submission, then you can submit it to test, and you don't have to worry that you're submitting something bad to the database, because it will be removed. And once you're confident that you do know what you're doing, you just remove that DEV from the URL, and submit it. So having registered a study, you next want to register a sample. Remember I said these describe the source of the DNA or RNA that you are using in your study. And samples have to conform to a checklist. So you can see I've listed a few of these down here. We have a human oral, human skin, human vaginal, microbial mat, plant-associated soil, wastewater, and there are many more. Either way, this is just going to give you a list of things or bits of information about your sample that you have to provide. Some of them will be mandatory and some of them will be optional, and there'll be a different set of mandatory and optional attributes for each checklist. But there's lots of different environmental sample checklists that you can choose from, so you just have to pick out the most relevant one to your data. And making sure you provide as much metadata as possible is going to make your submission much easier to understand for other people in the future. So submitting a sample interactively, you would then be presented with a web form which would be populated with the set of attributes that belong to the checklist that you suggested. But you'll always have a unique name, a title, a description, and details of the organism. So obviously with your sample, you have to specify what taxon you're submitting to, and there's lots of different special metagenomic taxa that you can use in your submission. So in this case, we've used the human gut metagenome as our example, and that has tax ID 408170. These come from a taxonomy database, so you have to submit something that is in the database, and this helps to make data more comparable once it's public in the database. So submitting your samples this way is convenient if you don't have many. You can fill in a spreadsheet as well and upload that to your browser, and that will fill out the form for you automatically. Or you can register your samples using XML documents in much the same way that I just described for studies. You fill out a template XML that we can provide based on the checklist that you've chosen. So I mentioned earlier that there's a slight complication when you're registering a MAG. You have to submit a special sample that refers to the metagenomic sample. So here's an example of how that might look. You include in this description line, this sample represents a MAG from the metagenomic sample, and then you put in the accession number of the metagenomic sample that you have previously registered. And then for your scientific name, you want to use an uncultured taxon. So this is going to indicate that it's some unknown member of this particular genus that you're interested in. And then the submission is simple from there. You just register your assembly to point to the relevant sample. So as far as submitting read data goes, this is going to need to be done using the Webin-CLI interface I talked about earlier. All you have to do is prepare your read files. So you can see we have here an example of a FASTQ file, and then you prepare a manifest file. This is a format that is developed for Webin-CLI, and it's just a list of values paired with a description of what they are. So for example, in this case, we have the platform value with the the platform tag with the value Illumina, the library strategy is clone end, the library source is other, and then we have references to the sample and study that these reads belong to. So this means that the database is going to know where to point the experiment and run at. And of course, there's the location of the FASTQ file. And you just send these things to ENA, and we process them, we validate them prior to your submission, so you know when you've submitted that everything in your submission is good to go. Submitting assembly data is much the same. You have here, for example, a FASTA file. So this is some assembly that we've done, and then you describe it in the manifest file once again. So we've got the name of the assembly, the coverage, the program that we've used to do it. And then you also include an assembly type option. It's not shown in this manifest file, but you include an assembly type option to indicate what sort of assembly you've done, be that primary, metagenome, bin metagenome, or MAG. But once again, it contains the kind of protocol info, the sample and study reference, and the names of the files or just file that you're submitting. and you send these to ENA using the Webin-CLI program, as I said. So we'll just take a moment to look at how you run Webin-CLI. So once again, you invoke the program by saying its name like this, Webin-CLI, and then you tell it what you're submitting. So the context option allows you to specify reads or genome or certain other things. And then you tell it where to find the manifest, which in this case is in test3 manifest.txt. And then you submit the files with the submit flag, and you give it your login data, so your Webin username and your password. Once again, this is test. We talked about this a moment ago with programmatic submission using XML. You can do a test submission using Webin-CLI if you're not confident of what you're doing, because obviously we can validate your files to make sure that they are valid FASTQ files, for example, but we can't make sure that you're making sure they point to the things you think you are. So you're welcome to test these things before you commit to a real submission. But that's about all there really is to submitting metagenomic data to ENA. We'll just run through that in overview once briefly. So the first step is registering a study. The second step is registering samples, environmental samples, and mag samples if you need them. The third step is submitting reads. And finally, you submit assemblies. So when you're ready to do your submission, you can register an account at the interactive submission homepage here. You can see tutorials, including a guide to metagenomic submission at our Read the Docs page. And you can get in touch with our help desk at this address here.